start. So the African Boxthorn Biocontrol and Drought Resilience Project is a national initiative led by CSIRO and supported by the Centre for Invasive Species Solutions and funded by the Australian Government Future Drought Fund. And apologies in the city, so if there's any alarms or cars, that's what's going on in the background. So Landcare Tasmania is in partnership with CSIRO, and we are working with the Tasmanian community and stakeholders to roll out the release of this boxthorn biocontrol in Tasmania between 2026 to 2028. And this will involve connecting with Landcare members, land owners and managers with CSIRO to release and monitor the biocontrol. We will also be running 3 field days across the state. In this webinar, we're also going to provide a update on the Sea Spurge community-led biocontrol project, which is again another national initiative funded by the Australian government's Department of Agriculture, Fisheries and Forestries. They're supporting communities managing pest, animals and weeds program. I will just note that this meeting is being recorded, so you can watch back, or you'll also be able to share it with any friends or colleagues. And biocontrol is just one tool in our toolbox for managing these weeds. We won't be going into further discussion around control of boxthorn in this webinar, but that's something that we will cover in our field days. So just a bit of a background on what we'll be covering today. So we are lucky to not just have one presenter from CSIRO, but we've got 3 experts in the room with us, which is great. So first we'll be hearing from Dr. Ben Gooden. So Ben is a principal research scientist and weed management system team leader with CSIRO. He'll be talking about the background of biocontrol and covering some of the history of weed biocontrol in Tasmania and talking a little bit more about the testing and science behind it. Then we'll be hearing from Dr. Gavin Hunter, who is a team leader of the Plant Health Diagnostics team and research scientist with CSIRO. Gavin will be presenting on the background of the Sea Spurge biocontrol project in Tasmania and Victoria. Then we've got Caroline Delassie, who is the Research Projects Officer with CSIRO, and Caroline will be presenting on community participation and engagement with biocontrol projects and how that will look for this project. And then myself, forgot to introduce myself. My name's Amy Henson. I'm the Southern Network Coordinator with Landcare Tasmania. And I'll be talking about the benefits of partnering with community Landcare and how Landcare Tas supports its members. We've also got Jen in the room with us, who's doing all of our back-end webinar stuff. She's our outreach coordinator. So you might see her. She's put herself, she's invisible. And also Anna Minchin, who is our project manager. Righty, so I'm going to hand over to Ben, and Ben is going to, I'm going to stop sharing.
Hello, yes, very happy to be here. I will share my screen.
Great. My screen was sharing that whole time, right?
It was. It looked fantastic.
Okay.
And
I'll put myself on mute now.
You may be able to hear that I'm a little bit hoarse, which is a bit annoying, and I'm sorry about that. So I'm going to mute quickly, have a big cough, drink of hot water, and then I'll be back to it. Okay, let's see how we go. And I will just check with you, Amy, before I kick off. Can you see my presenter full screen view?
Yes.
Excellent. Well, I'm very grateful to be here, and on behalf of the CSIRO team broadly, we are very pleased to be here to represent our organisation's research and relationships with community, but in particular, we're very grateful to Landcare for hosting today's webinar. We are increasingly engaging with community-led projects to intersect with national scale research led by us. The impact of the work that we aspire to deliver can only be delivered through building grassroots partnerships and relationships with community across all the diversity of stakeholdership. And it is truly a pleasure for us to be working across so many different projects with Landcare. We have engaged with Landcare on many different projects in different ways, where those relationships have evolved and emerged somewhat dynamically. These are the first set of projects where at the front end of project planning, we have had the intent to engage with Landcare. And so I think we've pulled that partnership off. And so we are launch padding with Landcare, and we're very grateful to be doing so. So thank you. And also to say this is actually our very first public meeting to launch this national scale project. So not just in a Tasmanian context, but also in a national context. So I think that's worth noting as well. So what I'll be presenting on today is more of a front end of weed biological control, the type of research that CSIRO does, the relationships that we have and that we build as part of that research pipeline. But of course, we are talking about biocontrol research as it is implemented in the context of Tasmania. And so two of the key weed species here, which are the quote, next cabs off the rank, so to speak, of novel biological control agents available to be released in Tasmania, where we need to work in partnership with community to implement that statewide, are for African Boxthorn, where the fungus has not been released in Tasmania yet, and Sea Spurge you will see in the bottom right-hand corner, which had 2 years worth of releases, but further funding has been made by the Commonwealth Government to expand that program more broadly. And before I kick off into the detail of my presentation, we too wish to acknowledge the traditional owners upon the land that we are calling in from. Gavin, Caroline and myself live and work on Ngunnawal country, the traditional country of the Ngunnawal people based around the Canberra region, and we also wish to pay our respects to their elders past and present. So my presentation really is broken up into two parts. The first speaks broadly about what do we mean for classical weed biocontrol? What is the historical context of this? It's important for us to do this because increasingly as we engage with grain growers and graziers and other production sectors, community-based conservation organisation, there are residual questions that are asked about, but is it kind of like a herbicide? And is it a bioherbicide that is regulated through the APVMA as if it was a chemical herbicide that you can go to Bunnings and purchase for you? And the answer is no. Classical biocontrol is delivered and regulated under legislation with a very particular framework for delivery. And it's important for me to give context to what that is and how we can work with it safely and how they can be implemented into works programs. Then I do talk very specifically about the African Boxthorn project. That's why we're here today, and we thank our partners for their support, the Centre for Invasive Species Solutions, who's coordinating the work. And then lead off with and finish off with current opportunities before I hand to Gavin to talk about Sea Spurge and Caroline to talk tangibly about how community can get involved in this. The CSIRO has a broad portfolio of research responsibilities. We're a Commonwealth Government organisation that tends to deliver research services to government, industry and community for weeds which tend to have more national scale impacts, many of which are weeds of national significance like African Boxthorn, that tend to have impacts across multiple different regions, state-based jurisdictions, across sectors. Jurisdictionally, we can cut across different state government agencies and jurisdictions to work in partnership. And our website has a full list of all the research projects we have. There are other organisations in Australia which are state-based organisations, Victoria, New South Wales and Queensland in particular do have dedicated research units for weed biocontrol. We partner with them and they do phenomenal work for their state-based priority weeds. So what do we mean by classical weed biocontrol? At a very simple level, it is introducing to Australia from the native range of where that weed is from naturally, what we term natural enemies of that weed. Typically, they are insects and fungal pathogens, as you can see from the example for fleabane that we're working on, where these biocontrol agents are co-evolved with the weed in the native range, which for flaxleaf fleabane happens to be Southern and Central America. And it is based on this idea that the plant, when growing in its native range, this is an example of a plant called Puccinia, which is from Argentina, that's invasive across Northern Australia, that in the natural context, the performance of that plant, the size of the population or its ability to reproduce and spread is regulated and constrained by being attacked by all of the co-evolved natural enemies, insects, pathogens, etc. And we tend to gather evidence for many of the weeds that we work on that when that weed has been introduced into Australia, it has left behind its natural enemies. In doing so, it is able to reallocate resources away from defending itself from attack into growing bigger, reproducing more often, spreading more quickly. And we tend to see this again and again. That forms the basis for decision making of, well, if we reintroduce, or rather introduce for the first time, these co-evolved natural enemies into Australia, can it put that pressure back on the weed to suppress its performance down to a much lower sustainable level? And again, for many of the weed species where biocontrol agents are available, this is what we see. Prickly pear, Patterson's curse, bidoo bush, their performance is reduced because they're being attacked by their natural enemies. This graph shows the weed population as the red line before a biocontrol agent is reduced in the blue line. And we certainly know in Australia that the environmental conditions that support weed abundance, that can fluctuate over time. So weed populations are not necessarily stable and static all the time. They do vary spatially and temporally. Then a weed biocontrol agent is introduced. The population of the agent does tend to expand following establishment very quickly. It plays catch up to the weed, and the biocontrol agent performance itself is benefited by having so much of the weed to attack. But then as the weed population begins to decline in response to biocontrol, they collapse down to a much lower level, which is more sustainable. And so it reaches a new lower level of dynamics. The weed populations are never removed altogether. The biocontrol agents may persist in the landscape at a much lower level, but the biocontrol agent is keeping the weed in check. And that may take the edge off to allow desirable native vegetation or other types of agricultural values to increase. So there are a few guiding principles that we lean upon for classical weed biocontrol. The first is that biocontrol agents are host specific. They have to have a narrow host range. If biocontrol agents have a broad host range and can attack multiple different plant species, including plant species that are quite distantly related to the weed, the regulations mean, through the risk analysis, that those biocontrol agents will not be released into the Australian environment. If they are released, they do tend to form self-sustaining systems. Once we get them established, and if the biocontrol agents can reproduce themselves from one generation to the next, just like the weed can do, the biocontrol agents do tend to persist and spread themselves in the landscape. There are a couple of examples, however, of where biocontrol agents may have limited dispersal, meaning that there is an opportunity for human assistance in spreading those biocontrol agents more widely. As Amy said, biocontrol is definitely not a silver bullet. There is no example anywhere globally of a weed biocontrol completely eradicating a target weed. And we tend to think of biocontrol as supporting more landscape scale weed control rather than site level weed control as a chemical herbicide would. Just check the timing, great. So in terms of the historical context, Australia really has led the way globally. We have more than 120 years worth of research practice in weed biological control, starting off in the early 1900s with Opuntia monacantha, which is an opuntioid cactus very similar to prickly pear with a cochineal insect that was released. Then the heyday of biocontrol, 1920s to the 40s, with the prickly pear situation, where the most successful biocontrol agent to date was released, the Cactoblastus cactorum moth. And let's remind ourselves how problematic prickly pear was, both for grazing productivity and now what we would understand as well as conservation of the woodlands that were invaded. 24 million hectares invaded. The moth was released. Now we can celebrate the 100 years of that program. And within a decade of that, it had cleared about 80% of the infested land that had otherwise been rendered degraded by prickly pear invasion. And in fact, the photographs for that program are now within the National Archives, and having a look through their website, we're starting to draw out those photographs and be able to undertake an analysis of the before and after impacts. This is a before photograph taken in the 1920s before the release. And then this is within a decade of release, the collapse of the infestation of prickly pear. So I need to highlight here that it is not a set and forget. Community collective action has always underpinned successful biocontrol campaigns. Even for prickly pear back in the 1920s and 30s, there is a direct quote here maintained in the Australian Archives that at the, the National Museum Archives, at the height of the operation, Chinchilla, which is where the moth was being reared, was sending out 14 million eggs of the moth a day across Queensland and New South Wales to farmers for release. It's just that more recently we have not established these broad national networks of community-based releases. So it's almost like we're connecting back into that best practice of community relationship that had been forged in the 1920s. Oh, excuse me. Okay, I'll move past that. So where is the situation now in Australia? There are more than 270 agents approved for release for more than 60 weeds. Across the weeds that could be assessed, for which there was sufficiently good quality data, the return on that investment is 23 to 1. Of course, I think it's worth noting that that return on investment is very much framed in terms of agricultural productivity. There are biodiversity and conservation gains, which are very hard to quantify, and there's an argument that it's not appropriate to quantify them in a return on investment type of context. And there is an ongoing need for further integration of biocontrol into practice. Biological control has a clear regulatory pipeline with multiple phases of research. Phase 1 is when researchers from Australia, in partnership with researchers based in the country of origin, work together to explore for different potential biological control agents of interest. If we find any of interest, we move to a phase 2 of research where we undertake a risk assessment in our laboratories to make sure they're safe. If safe and approved for release, we move to the mass rearing and release phase, phase 3, which is where we are at now for African Boxthorn. And then coupled with that in the very long term, monitoring and evaluation of how successful or otherwise the programs of work have been. I'm just going to take a sip of my cup of tea. African Boxthorn. I suspect there are many people on the call today who were drawn to this conversation on the basis of an interest in Boxthorn. Also many for Sea Spurge. I will now be talking specifically about Boxthorn. It's native to South Africa, one of the most pervasive woody weeds across southern Australia, and it's in the mix with some pretty wicked other ones, bitterbush, boneseed, gorse, blackberry, they are especially recalcitrant to control and very hard to control with biocontrol. Significant agricultural impacts, but in addition to that, also significant conservation impacts. There are some wicked management challenges. We are working in partnership with the New South Wales government, and we have integrated management sites where we are controlling boxthorn with many different methods and following up how it's performing. Well, these photographs have just come in from my colleague Blake from the New South Wales government, that off the basis of cut and paste and chemical herbicide application, that those plants, they're so resilient to being controlled mechanically and chemically, rapid resprouting. And even when there was really good foliar application of chemical herbicides, they are resprouting this was within the space of 6 months of control. And there are other challenges too. Even if the chemical herbicides work well, and they do in many contexts, there's restrictions around how they can be used in an ecologically sensitive way, if it's along a creek line or in the context of endangered ecological communities that are grassy woodlands. And also the birds disperse the fruit of African boxthorn very quickly. And even if you can get rid of all your boxthorn, if you have a nice roost tree for birds that are frugivorous, they will fly back into those roost trees and defecate out the fruit, and the infestation of boxthorn starts again. So we're looking for other complementary methods to break the back of that cycle. So in terms of exploratory research for the fungus, work was done by colleagues of ours, led by Dr. Kylie Ireland, who is no longer at CSIRO. She's based in Western Australia now. She undertook expeditions across Southern Africa to identify different populations of boxthorn and have a look at all the different insects and fungi that were attacking it, and identified what looked to be a pretty good fungus, a rust fungus called Puccinia rapipes. And I've put these images up to show that there's a lot of front-end research that goes on to make sure that we're selecting the fungus from the plants that are most closely related to the ones in Australia. If you get the fungus from the wrong type of boxthorn, then there's a genetic incompatibility, and the fungus, when released over here, may actually not infect the weedy populations as much as we want. And also that we must understand what the fungus is, how does the fungus reproduce, complete its life cycle, and we also do genetic analysis so that we can understand exactly genetically what the strain of the fungus is that we're working with. And here it is, Puccinia rapipes. It is a classical rust fungus. The spores are released, they land on the leaf, they infect the leaf tissue, causing this speckling. They complete their life cycle over a few weeks. Once the life cycle has been completed, like when you go bushwalking in autumn, you can see toadstools emerging from the soil. All of the fungus is in the soil. In this case, most of the fungus is in the leaf. And just when they reproduce, you can see all of these little speckles emerging from the leaf. That's the fruiting body emerging from the leaf, full of all the spores that are released to go on to infect another nearby plant. And then at the end of that cycle, the leaves that are infected shrivel and die off. The host specificity testing was undertaken, in fact, by Gavin and Kylie. And Gavin is in the foreground in this photograph. He's now working on Sea Spurge, but he worked with Kylie on this plant too. And the host specificity testing is used by exposing the fungus to a whole bunch of native and ornamentally and agriculturally important plant species that are at most risk of infection, and they tend to be the ones most closely related to African boxthorn. And on the basis of that testing, an independent risk assessment determined that the fungus was safe for release into the Australian environment. And that's what we've done in New South Wales. So we've actually had 2 years worth of trial work in New South Wales in the lead up to this project. There are many lessons we've learned from this. I'm just going to check the time. 17 seconds till my 20 minutes. I might indulge myself with a 30-second extension if that's okay. The fungus can establish.
Sorry, Ben. Your slide is I'm not sure if it's for everyone else. It's sort of halfway between. Oh, there we go. That's better.
Got it. Good. The fungus can establish in the Australian environment. That's a really important milestone for us because up until release is approved, we can only undertake epidemiological and infection research in the laboratory, not in an ecological context. So when we released it in New South Wales, we have monitored, can it establish? Well, the answer is yes, it can. Can it establish and persist across multiple years? Yes, we've identified it can. These were in fairly harsh environments. We anticipate that if the fungus can establish well in the central west of New South Wales, where the previous 2 summers were particularly challenging, we see no reason why the fungus would not establish well in Tasmania. We have not found any evidence that infection by the fungus causes defoliation of the plant. So it is not going to kill the plant in the short to medium term. We are fairly sure of this. But within 12 months, we did find if you are a stem infected severely with the fungus, you on average produce 62% fewer fruit than comparable stems which are not infected by the fungus. This is a real win for us because if you consider the management challenges for boxthorn, as exemplified by this riparian corridor, which is covered in African boxthorn at a site that we're working at, that cannot be chemically controlled. The conservationist and the farmer who are controlling boxthorn are doing so in the paddock trees up here. Well, the fruit just keep coming in again and again and again. So what we're saying is you can actually integrate the fungus spatially by releasing it in the creek line, getting it well established, reducing how much fruit is produced, which at the landscape scale may in fact reduce the invasion pressure. So I'm going to finish up now, and thank you for letting me talk a little bit longer than I had planned to, but I do run away with myself occasionally. To acknowledge that this is not just a Tasmanian program. Tasmania forms a really important hub within truly a national network of multiple agencies working across all state and territories for multiple weed species targeted for drought resilience. All of these species have some sort of significant impact on ecosystem and agricultural drought resilience. By reducing their impact, we seek to enhance drought resilience. Funding from the Australian Government through the Future Drought Fund, coordinated nationally by the Centre for Invasive Species Solution, then delivered on ground by each of these research partners. So thank you again, highlighting the critical importance of community collaboration. All parties are invited to participate, and we work with each jurisdiction and person on an individualised basis to determine how best you can release the biocontrol agent with our support. So I'll finish there. Back to you, Amy. Thank you.
Thank you, Ben. That was, I learnt a lot. Thank you so much. Now we're going to hand over to Gavin, and he will talk about Sea Spurge.
Thanks, Amy. I'll share my screen. There we go. Can you see that?
Yep.
Great. Excellent. Evening, everyone. My name is Gavin Hunter. I'm a plant pathology research scientist based at the CSIRO here in Canberra. I've been working together with Ben and Caroline on the biocontrol of Sea Spurge in Australia. And I'll provide an overview on kind of the discovery of the biocontrol agent, its development, and its eventual deployment in Australia. So, the CSIRO, with co-funding from the then Tasmanian Department of Primary Industries and Water and the Australian government, undertook surveys in France for any pathogens and arthropods of sea spurge during 2008 to 2009 to try and find possible candidates for classical biocontrol of sea spurge in Australia. During the survey, 22 sea spurge sites along the Mediterranean and Atlantic coast of France were sampled, and these surveys resulted in the collection of 4 insect species and 2 fungal species. Now, one of the fungal species produced these really impressive disease symptoms on sea spurge that you can see on the right-hand side there. This specific fungus was collected from 9 of the 22 surveyed sites in France, and after some initial host testing with this fungus at the CSIRO European Laboratory in Montpellier, it was decided that this fungus was a promising candidate for further host specificity testing in Australia. So to undertake host specificity testing for candidate biocontrol agents, we have to first develop a comprehensive host test list of non-target species. This is done by using a technique known as the centrifugal phylogenetic method that is based on the phylogenetic relationships of test plants to the target weed. And the method is really underpinned by evidence that specialist candidate biocontrol agents are evolutionarily more likely to infect non-target species that are closely related to the target weed relative to those that are more distantly related. So for Sea Spurge, we investigated published literature for the family Euphorbiaceae to develop a list of related euphorbia species to include in host testing experiments with the biocontrol agent that was collected in France. The final host test list included 44 non-target species from the families Euphorbiaceae, Picarodendaceae, and the Phyllandaceae. And our Sea Spurge is accommodated in subgenus Eschula within the genus Euphorbia, and we were able to include about 19 non-target species from the subgenus Eschula in the host testing. So overall, we managed to get a really good representation of the phylogenetic breadth and diversity from the species that we included in the final host test list. In 2017, the New South Wales government, through the New South Wales Environmental Trust, provided co-funding with CSIRO to undertake host specificity testing with the candidate biocontrol agent of Sea Spurge. This funding allowed us to develop mass culturing and release techniques for the fungus. And the fungus was imported into the biocontainment facility at CSIRO here in Canberra, where we proceeded to test if the fungal agent was specific to Sea Spurge. Essentially, we would mass culture the fungus and in these growth chambers, and then harvest the fungal spores to prepare spore suspensions, which were sprayed onto non-target Euphorbiaceae species from the host test list and onto Sea Spurge plants that acted as positive controls. Following inoculation, the plants were assessed over a 30-day period to identify any signs or symptoms of infection. During this time, we also took leaves from inoculated plants and viewed these with microscopy to determine if and how the fungus infected and developed within the inoculated plant species. This table indicates some of the results from the host specificity testing and the microscopic observations. You'll see the furthest column on the right-hand side specifies the plant response to the candidate biocontrol agent. For this, we determined the 4 response categories, namely highly susceptible, resistant, and immune. They were based on visible symptoms observed on the plant and the microscopic development stages of the fungus on and inside the plant. The take-home message here is that Sea Spurge, or Euphorbia pyralius, is highly susceptible to the fungus, and the fungus was able to penetrate Sea Spurge leaves, grow within the plant, produce reproductive structures and spores, and complete its life cycle on Sea Spurge plants. You'll notice that all the other species, except for Euphorbia sagittalis, were either characterized as immune or resistant to the fungus, meaning that the fungus was not able to penetrate and grow inside the plant, and the resistance of the plant to the fungus kicked in at those levels. So once all of the host specificity testing with a candidate biocontrol agent was completed, all of the data and results were compiled, and an application to release the biocontrol agent into the Australian environment was submitted to the Australian regulator, who proceeded to undertake a comprehensive risk assessment of the data and results that were presented in the application. Once the regulator had produced a draft risk analysis, it was submitted to the Plant Health Committee for their feedback and also underwent a public consultation. After this whole process, we received notification in November 2020 that the regulator recommended the release of the fungus for the biocontrol of Sea Spurge, and that the overall risk associated with the release was negligible and that it achieved Australia's appropriate level of protection. Further co-funding from the New South Wales Environmental Trust from 2021 to 2024 saw CSIRO embark on a project focused on releasing the biocontrol agent along the coastline lines of Tasmania and Victoria. As part of this project, the CSIRO project team collaborated with Parks Victoria, Tasmanian Parks and Wildlife Services, and the Sea Spurge remote area teams, SPRATs, particularly Dr. John Marsden Smedley, to identify and establish 9 long-term monitoring sites, so 6 in Victoria and 3 in Tasmania, along the coastline at Sea Spurge infestations. Each monitoring site consisted of 3 10-meter long transects, and each containing 5 1 square meter plots. The biocontrol agent was sprayed onto sea spurge plants in each plots, and the plots were monitored at roughly 6-month intervals over a 4-year period to obtain quantitative data to inform the disease incidence, foliage health, foliage cover, and reproductive output of inoculated sea spurge plants. This was one part of the project, and the other important part of the project was community-led releases of the agent across Victoria and Tasmania. So from active and in-depth observations of the biocontrol agent in the field over the course of the past 4 years, we've been able to determine the epidemiology of the agent and the disease it causes on sea spurge. My colleague Caroline has developed this informative pictogram which details the progress of the disease in a field context. So firstly, the biocontrol agent penetrates sea spurge leaves, and infection results in these large, dark brown, concentric lesions. The leaf lesions generally increase in size, and the pathogen will eventually move from the leaf into the stem of sea spurge. Once inside the stem, the biocontrol agent will progressively girdle the stem and form these characteristic dark brown stem lesions extending up and down the stem. Now, because of the stem infections, the sea spurge plants, often their ability to efficiently transport water and photosynthase is reduced, which eventually causes the stem to topple over at the point of the stem lesion. Or alternatively, you may also observe that the tops of the stem above the stem lesions start to die back. This is an overall picture of the infection process and symptom development in the field and provides a helpful guide when looking for disease symptoms. Let's just go back. So as mentioned, we regularly return to the 9 dedicated sea spurge monitoring sites across Victoria and Tasmania to gather various data to inform us on the establishment and the initial impact of the agent. One of the measures we're interested in is disease incidence, which is the percentage of sea spurge plants per plot exhibiting leaf and stem lesions. We observed that there was a strong temporal variation of leaf lesions on sea spurge caused by the biocontrol agent, with a sharp increase in the percentage of plants showing leaf lesions in late 2022. In contrast, for stem lesions, it was clear that there has been a steady increase in the percentage of sea spurge plants exhibiting stem lesions over the 4-year assessment period. Excitedly, from all of the various assessments that were undertaken, we can confidently say that the biocontrol agent was detected at all of the 9 monitoring sites across Victoria and Tasmania, and as such, we can conclusively say that the biocontrol agent has now established well in these areas where it was released. One other indicator that we were interested in observing was the foliar health of sea spurge plants in the plots where the biocontrol agent was released. When the agent was first released back in late 2021, there was initially a low disease severity. However, this progressively transitioned towards a high disease severity to a point where we were able to determine that there was an overall 53% decline in sea spurge foliage health over time at the monitoring sites where the agent was released. So again, very positive news that the sea spurge is being impacted by the agent at these monitoring sites. An important point to make too is that the biocontrol agent, once released and established, will begin to naturally move and disperse to other sea spurge plants in the area. If the prevailing environmental conditions are conducive, the agent will sporulate and produce many spores on the leaf or stem surface, which will be blown to other sea spurge plants and initiate new infections. As a result of this, the agent will progressively begin to spread along the beach, and we've witnessed this movement during our assessments of the agent at the monitoring sites in Victoria and Tasmania. In Victoria, the agent spread from all sites, and you can see in the first photo plate over here of 5 monitoring sites in Victoria of the dispersal of the agent. The yellow star is the monitoring site where the agent was released, and the yellow lines demarcate the area where the fungus was identified during 2024. And in the case of these sites, we estimated that the minimum dispersal was approximately 250 meters, while the sites that the agent dispersed, or other sites where the agent dispersed, up to 1,000 meters away from the original release point. A similar situation was observed in Tasmania, where the agent had naturally spread at all 3 sites, up to a maximum of approximately 350 to 400 meters away. This picture on the far right, which was prepared by our collaborator, Dr. John Marsden-Smedley from Sprats, also shows that the agent was able to hop over an area with little or low sea spurge abundance to a different sea spurge infestation further down the beach. Just to share some of our latest analysis results with you, specifically for 2 of our long-term monitoring sites in Tasmania, we've now collected data across 7 monitoring time points of sea spurge plants inoculated with the agent over a period of about 5 years. And analyzing this data, we've been able to show that sea spurge foliar cover significantly declined over that time period following release of the biocontrol agent. At the 2 release sites in Tasmania, foliage cover declined by 19% from an average of 31% at baseline assessment to 12% at 4 years following biocontrol agent release. Our analysis also indicated that there is a significant effect of time since release of the agent on sea spurge and its reproductive output. Sea spurge reproductive effort was on average of 54% at baseline assessment in 2021, and this decreased to an average of 26% 4 to 5 years post-release of the biocontrol agent. So continued positive news in terms of the effect of the agent at some of these monitoring sites in Tasmania. I'd also just like to give an overview of the results from some of the community releases of the biocontrol agent. So the agent was released at a total of 231 sites, 128 of these sites in Victoria and 103 sites in Tasmania. Across all of these sites, the agent was detected at 61% of the monitoring sites by the community members, which is really great news for us. The releases were undertaken all year round, sometimes in less than 25 degrees Celsius, and the agent was also detected all year round, but mainly in spring. And finally, I'd like to touch on just a few points regarding the future monitoring and research directions on the current project. The agent is now well established in Victoria and Tasmania, but sea spurge is distributed across the entire southern coastline of the country. As such, we'd like to facilitate a national scale release of the biocontrol agent together with community participation so that it can be released at as many sites across the entire distribution of the sea spurge along the southern coastline. Also, in collaboration with enabling partners, we'll be establishing further long-term monitoring sites in Western Australia and South Australia, where we aim to undertake similar dedicated monitoring as we did in Victoria and Tasmania. We're also very keen to collaboratively co-design possible release programs with Indigenous communities and Indigenous ranger groups for release of the agent on country. And we plan to collect as much quantitative data to provide information on the agent's effectiveness and use this information to refine best practice guidelines for eventual integration into weed management programs. So I'll thank you for your time, and I've just provided some web addresses there that you can go to in terms to get some more information on the Sea Spurge Biocontrol project and also to register your interest in participating as a community. Great, thanks very much.
Thanks, Gavin. Thank you. And these links will be shared in a follow-up email. So if there's, and as well as the presentation. So if there's anything particular that you missed or you want more detail on, that will be in the follow-up email. Okay, now we'll hand over to Caroline.
Hi, everyone. I'm just sharing my screen. Hopefully that comes up. Oops. There we go. So hi, everyone. I'm Caroline Delace. I'm sure some of you may recognize my name from some of the emails you may have received from me around both the Sea Spurge and the Boxthorn biocontrol programs. But today I'll be talking about the community involvement, specifically more so for the Boxthorn program. So some of the main key things that we often have an importance with the collaboration aspect is that so some of the points just listed here around leveraging local knowledge to identify target weed populations and ecological nuances. For an example, we often use ALA or iNaturalist to identify weed populations. However, often a lot of these populations in remote areas are not typically recorded, as well as identifying populations that may be on private land as well, and getting access to those populations that typically would not be controlled. We also hope to integrate community feedback into these programs by combining the science with practical on-ground experience, as well as integrating biocontrol into current control strategies that are already in place. We also hope to respect the local knowledge and cultural practices, especially that of the First Nations, and aligning biocontrol efforts with also the biocultural values of those areas. We also really importantly hope to create opportunities for knowledge exchange and also capacity building across sectors. So we want long-term sustainability of these biocontrol programs by training stakeholders on how to release the agent, monitor and collect data, especially once these projects come to an end for us. We want the community to be able to keep monitoring the biocontrol agent and hopefully assess its spread over time, as well as lodging this through databases that we hope to set up in the future for other people to refer to where the biocontrol agent is present. We also would like to build trust and shared ownership of these biocontrol programs. Having that transparency and inclusion allows for that improved uptake of biocontrols and facilitates smoother regulatory and operational processes. And like Ben mentioned, we also really want to reduce chemical use and improve ecosystem health, especially in those areas that are typically not targeted for weed control, and build a stronger community resilience. So this is sort of what the process would look like when you participate in the program. I'll go through each step with you in this presentation. So starting off, we typically do things like what we're doing today: webinars, workshops, presentations, media releases, and so on. But I'll focus a case study on the New South Wales Trust project that ran between 2023 and 2026 and how that outreach could look like. So this is a map basically of the releases that happened in New South Wales for the Boxthorn biocontrol program. And this sort of just shows that sort of widespread distribution across New South Wales. But in particular, I'll be focusing on this region in Hay or Deniliquin area. It's all part of the Riverina in New South Wales. So some of the key things that we found that were really helpful was having forums and workshops in person, such as those images on the right there. These are just some examples. These were all run by the local land service in New South Wales. We also found that the local government did a lot to work to register land managers. This was in particular to assist those who had limited email or internet access due to just not being on things like Facebook or online to see these events being run, as well as so part of the delivery of the biocontrol agent is through post. So some land managers may often not have access to post, or they would have to drive an hour into town to get access just due to their remoteness. So what would happen is they would have a distribution point, which they could all go to, collect their biocontrol agent, and then head back home later. They also distributed a lot of information to land managers who were unable to attend workshops, whether it's face-to-face meetings or phone calls, which then goes on to the next point, which is where often then they could pass on our contact details to land managers or those controlling their boxthorn populations. We also found that was really handy and quite useful that land managers actually worked together. So they would often, almost like a network, work with each other to organise a day where they could all release the biocontrol agent together, help each other around, as well as assisting neighbours who perhaps didn't have the time or ability to make the releases themselves. So as a result, we actually had quite a huge uptake of this biocontrol agent in that area due to these reasons. So part of the next step after workshops and presentations, we basically will register the participants. So there's 2 different streams for this particular project with African Boxthorn. You can register via Landcare Tasmania, which Amy will probably talk more later about, or through the emails, as well as other information. And anyone who is a non-Landcare member can just register through us. So part of your registration process will look similar between registering through Landcare Tasmania or registering through us. We'll just have a few information sheets for you to read through, in particular the biocontrol agent information sheet that goes into detail about the biocontrol agent, what to expect, what to look for in a site, and so on. The second part is the research information sheet. This is around human ethics, just how we will manage personal data and that it is fully confidential and will not be shared with anyone else. The second part, once you have read these, is registering through our either online form or physical form. And on the right, here's just an example of the Landcare registration form that's online as well as ours. Some things when you register is it will request things about your nominated sites that you would like to release at. When selecting your sites, we typically recommend that you choose sites that you're not planning to control, as well as looking for areas where you have healthy boxthorn plants with green foliage, as that is usually the best way for usually the healthier the plant, the better the biocontrol agent will establish and survive. From there, once you've registered, we will then post the biocontrol agent to you, along with the release instructions and some data sheets. So this is what a kit typically looks like. We'll provide you with a small vial of spores, small but mighty. It doesn't look like a lot, but it goes a long way. So that will be enough to, basically one kit will be enough to release across 8 to 10 boxthorn stems. So that can be across 8 different plants in an area. From there, that, as Ben mentioned, and Gavin as well with the Sea Spurge project, typically the way a biocontrol agent works is you release a small amount, but that will gradually spread across to neighbouring plants. By releasing less at the start also means that you're not spending hours spraying every single plant. So it's less effort as well for the participants. We also provide plastic bags, but I will explain that what that is used for in the next slide. So the next step is then for participants to make the releases. So the way the biocontrol agent is released is the spores are mixed in with tween, which is a surfactant, and some water. That is then just sprayed onto the branches, and then that is then covered with a plastic bag. Super easy, but also a little bit difficult with some of the spikes, but the bags themselves are quite tough. So we're hoping that that should hopefully hold very well. We typically recommend making the releases in autumn when the temperature is quite mild, as well as being later in the afternoon. With those plastic bags around the branches, that will create a greenhouse effect. So if you've got the sunlight going through the bags, it will actually heat up inside the bag, which will actually more often than not kill the spores. So we definitely recommend doing it as late in the afternoon as possible. Upon making your releases, we've just got a few data sheets to complete. They're very basic level data that we're basically collecting, just saying where did you make your releases, time and date, number of kits released, and how many branches you sprayed. Post-release, so say 10 weeks or in spring, we've got a few other things for you to collect. That is just around, did you see it or not? How widespread is it? So really simple data to collect, but it is very informative for us to know. So when it comes to monitoring, this is typically some examples of what we look like here on the left. These small little bumps that we call pustules, they form on the leaf when the when the fungus is basically at its healthiest, typically during spring, when the weather is nice and warm and optimal for the spores. From these pustules, they will release the spores into the air, which will then go on to infect new plants. Of course, there are lots of other things on boxthorn, so that does make it a bit tricky, such as lichen or white marks from, say, thrip. These are not related to the fungal biocontrol agent. These are completely different things. However, if you're ever uncertain, we we are more than happy to look through photos that you send us, and we can confirm that with you. Some important notes, as I've mentioned, releases are made in autumn. Monitoring can be done 10 weeks after or in spring, depending if it's super cold. Typically, the fungus can lay dormant in the leaves, and then it will come through in the spring. As Ben mentioned, Ben also mentioned, impacts on boxthorn is not expected to happen short term. It's usually long term. And we do not expect the fungus to kill the host plant, but it will reduce health and reduce the seed set over time. It is basically a complementary tool to other control methods, as Ben had also given examples for. And at the end of that, all that data will come together, and we can basically form nice maps like this one here, where we can see the dark grey spots indicate where all of the records are on ALA for African Boxthorn, and we can see that we've got a really nice spread of where the biocontrol agent has been released with those green points, as well as we can start mapping out where people have seen infection versus where they haven't, and start to see if we can correlate that to any environmental conditions that may be present. But as you can see at the moment, we've got quite a nice spread of infections present that has been recorded across New South Wales. So here's some of the contact details about if you want to get in contact more about the Boxthorn program. And yeah, that's all from me. Thank you.
Thank you, Carolyn. Alrighty. Thanks everyone. That was really informative. And it's really exciting for us at Landcare to be partnering on such an innovative project. So I'm going to now, hopefully that's sharing. I'm going to quickly just give a talk about Landcare Tasmania and also just about the benefits of partnering with community Landcare, which we have sort of heard a little bit about the benefits from our previous presenters. So apologies if there's any kind of double ups, but we'll see how we go. So Landcare Tasmania. So we are the peak body for community landcare in Tasmania since 1994. We're a not-for-profit that supports community members to achieve their landcare goals. And we are unique in that the fact that our members are the heart of our organisation and we are led by them. So who are these members? So as you can see there, we've got a map of our member groups from that was updated in 2025. So we are across the whole state, and we're made up of community care groups, school groups, associates, individuals, and over 8,000 volunteers, which is pretty cool. And so why is this project important to our members? So we know from last year's membership data that 67% of our members mentioned weed control as one of their key key landcare activities. Apologies, I think I'm going into shadow because the sun is setting. And currently there's 14 projects in our project bank, which I'll talk a little bit more later, for members wanting funding to manage Boxthorn. So Ben has spoken a little bit about Boxthorn more generally, and that it's a weed of national significance. This slide is going to just talk kind of high level what that means and what that means in Tasmania. So under the Tasmanian Biosecurity Act 2019, it is a declared weed. So all landholders have a responsibility to manage and prevent spread. The legal status of Boxthorn in your area is determined by the state statutory weed management plan, which identifies management zones. So there's two management zones, Zone A, which is eradication, and Zone B, which is containment. So the current zoning as of 2011 is Zone A includes the Huon Valley, the Tasman, Kentish and Devonport. I will also note that Biosecurity Tas is in the process of updating statutory weed plans into a new biosecurity weed program. And this map that you can see here is from the Naturals Values Atlas. It's a map from the past 20 years of boxthorn records. So you can see that it's all along, all around the state, even in King Island and Flinders. And it's just wanted to highlight that these zones, are kind of determined by the distribution of records that are accessible. So the more that we input our records of prevalence of weeds and species, the better, I guess, our zoning and planning can be. So what are the benefits of partnering with community land care? So I'm just going to give a case study or an example of a community care group that has achieved long-term environmental outcomes. And while it's not a story about biocontrol, it's still a good example of what groups can achieve. So the Port Signet Land and Water Care Group was established in 1995, and it's worked across multiple land tenures and has partnered successfully with the Huon Valley Council, as well as engaging with 38 private landowners and the broader community. They focus on willow removal, and they've seen 7 kilometres of riparian area, that the area in the blue on that map, restored or revegetated in Signet, which is in southern Tasmania. They've also received several grants from Landcare Tasmania and also the Huon Valley Council. So the benefits of partnering with a community land care group, like on a project like this, is and Carolyn has already kind of touched on these on this, but so community buy-in and ownership is a big part of project success and longevity. So having support from landowners and the community means that they will provide ongoing support, ongoing monitoring and maintenance and follow-up of sites and weeds. You're also going to get peer-to-peer learning and sharing. So addressing funding and resource constraints. So our groups have a governance infrastructure to be able to apply for a range of grants and funding, and they provide thousands of volunteer hours for on-ground work, for project management, and also for admin. They provide a good return on investment for grants and other funding and support. Local expertise. So land care groups can strengthen the capacity of the local community by sharing and providing guidance from those local experts and those with long-term experience. And finally, and not completely, but landscape-wide and community impact is another a power of our land care groups. So as Carolyn said, working on different land tenures, community has access to private land and also works on parks land and council land, and they also engage with lots of different stakeholders and community groups. They provide a framework for local land owners and communities to make plans for landscape scale projects, which increases the scale of impact. So what is Landcare Tasmania's role? So we provide the next governance and support infrastructure for all of our Landcare members across Tasmania. So what that support might look like, so that's a list of our key programs. But some key things to note is that we provide access to insurance and governance support. We provide access to the project bank, which is where our members can submit a expression of interest for a project idea for funding opportunities. We run capacity building workshops and educational events like what we're doing with Boxthorn. We provide access to resources and newsletters. We connect our members with networks and stakeholders. We provide groups their own logo, so they can get their own Landcare logo, their own Landcare email, as well as Google storage. We also provide project support. We have a lending library of equipment, and lots more that you can read on our website. So how to get involved in this project? We've heard a little bit from Carolyn about the different or the kind of the stage and the process of being involved in this project. So if you are a Landcare member, you can use this QR code that will take you to our form. If you're not a Landcare member, you can also use this form and it will direct you. There's a link that will direct you to CSIRO's website. We currently have 11 entries in our form, so thank you for everyone who's already put in a submission. So some things to remember when you are applying, if you are not the landowner, you must have permission from the landowner. So if you're working on parks and wildlife managed land, that would look like contacting your regional office and lodging a works and development application request. If you are working on council land, then we advise you to contact council and request a letter of permission. So you can be any type of Landcare member. So you could be a group if you wanted to release it as a group, and you can also register multiple sites. You'll receive multiple kits. So in the form, it's going to ask you a few questions about the size of the infestation and then just the GPS coordinates of where that infestation is. Also, as I mentioned earlier, part of this project is that we'll be running some field day demonstration sites with CSIRO, so they will be coming down and running those with us, which is really exciting. We'll also have a local weed expert joining us to talk about follow-up physical and chemical control as well. So we've got our dates locked in. We are still confirming our locations. So the south will be on the 22nd of April, the east will be the 14th of May, and the north will be the 1st of October. If you're in the east or the north and you have a decent infestation of boxthorn and you would be interested in hosting a field day at your site, please let us know. And finally, if you've got follow-up boxthorn control projects that you would like funding for, you can always put that into the project bank as well. So that's always open for you to submit a project. Great, that was just on time. So again, I would like to thank everyone for joining us today and for our speakers. It's been just fantastic experience working with CSIRO, so we're very grateful, and we're very excited to see our members already submitting their sites for who are interested in the release. So as I said, we will be sending a follow-up email that will have this recording and links to follow up. And the email address is of CSIRO if you would like to get in contact. So we've got about 15 minutes left. I'm not sure if anyone has yet put a question into the chat.